Hello everyone. So the next topic which we will be going to discuss is about the chemiosmotic hypothesis. So let's start with the introduction first. So what was the history of chemiosmotic hypothesis? In 1961, Peter Mitchell postulated this hypothesis. So what he explained? He explained the mechanism of ATP synthesis with chloroplast during the photosynthesis process. So he gave the mechanism of synthesis, ATP synthesis with the chloroplast. So during the photochemical phase of or light energy, ATP and NADPH are generated. So when there is a light reaction in light reaction is also a part of photosynthesis. It is subdivided into two parts, dark reaction and light reaction. So in case of light reaction, ATP and NADP are generated. So they are the key components and they are used in the dark reaction for the production of the final product of photosynthesis, that is the sugar molecules. So in case of light reactions, it is the ATP and NADP, and in case of the dark reactions, it is the sugar molecule which is the final product of the dark reaction is the sugar molecules. Now this is a diagram which is representing that how this chemiosmotic hypothesis was being postulated. So as per the chemiosmotic hypothesis, ATP production is the outcome of proton gradient which is established across the membrane of the thylakoids. So thylakoids, they are present in the chloroplast. So ATP production is the outcome of the proton gradient. So here the first is being labeled as the inner mitochondrial membrane. Second is the intermembrane space, and third is the outer mitochondrial membrane. And there is a plus sign which is outside of this inner mitochondrial membrane, and inside of this there is a low H plus ions, and outside there is a high H plus ion. So ATP production is the outcome of the proton gradient which is established across the thylakoid membranes. So the required components for chemiosmosis are proton gradient, proton pump, and ATP synthase. So these are some of the required components. Now we will discuss what is the function of ATP synthase here. So ATP synthase, as it has been an enzyme because it is a synthase, it is an enzyme that aids in bringing about ATP synthesis. So there is a synthesis of ATP, and the enzyme which helps in the synthesis of ATP is the ATP synthase. The enzyme has two portions, that is F0 and F1. So there are two portions, F0 and F1. So what is F0? F0 is a transmembrane channel, while configuration changes in F1 activate the enzyme. So both the F1 and F2 portions are being depicted here, that it is a transmembrane channel, while the configurational changes in F1 activate the enzymes. So they phosphorylate the ADP, and one of the driving factors of ATP synthase is the protein gradient that is developed across a membrane. So ATP synthase is the protein gradient that is developed across a membrane. Now we will discuss more in detail about the chemiosmotic hypothesis. So what happens is, in the case of light reactions, photosystem, they help the chlorophyll to absorb the light. So it is the chlorophyll which helps in the absorption of light. So there are two kinds of photosystem, photosystem 1 and the photosystem 2. So the overall role of the photosystem is to absorb the light with the help of chlorophyll. So as a result, hydrolysis, which means the splitting of water, takes place and releases the electrons and the protons. So they will release the electrons and the protons. In case of electrons, electrons get excited to a higher energy level and they are being transported to the electron transport system, while protons, which is in hydrogen ions, they are from the stroma, they accumulate inside the membrane. This creates a protein gradient, and this helps to create the protein gradient, and some protons are used by photosystem 1 for the reduction of NADPH. So they are responsible for the production of NADPH. So when a proton gradient is collapsed, so when the proton gradient is collapsed, it will release the energy that was being accumulated, and it will release the protons that were accumulated inside the membrane. So they carried out back to the stroma via the F0 phase, which was the transmembrane channel of ATP synthase. So the released energy causes changes in the F1 stage. So they will cause the change in the F1 stage and triggers the ATP synthase to convert the ADP to ATP. So there is an energy production. So now what is about the chemiosmotic theory? So chemiosmotic theory is about the molecules like glucose. They are metabolized to develop acetyl coenzyme A in the form of an intermediate that is energy rich. So the acetyl coenzyme A is in the form of an intermediate that is energy rich. The proper oxidation of acetyl coenzyme A occurs in the mitochondrial matrix, and it is combined to the reduced form of a carrier molecule, namely FAD and NAD. So this is the proper oxidation of acetyl coenzyme A in the chemiosmotic theory. The carriers then supply electrons to transport the chain of the electron in the inner membrane of the mitochondria, which further supply them to a different other proteins present in the electron transport chain. So this energy which is present in the electrons will basically use to pump out the protons from the matrix in the inner mitochondrial membrane, and it is used to store the energy in the form of a transmembrane electrochemical gradient. So the protons, they return to the inner membrane by the ATP enzyme synthase, and the proton flow travels from the into the matrix of the mitochondria through ATP synthase, which will get a good amount of energy for the ADP to integrate with the inorganic phosphate to produce the ATP, that is the energy. So this was about the chemiosmotic theory, and we have discussed the chemiosmotic hypothesis. Now what are the evidences which leads to the process of the chemiosmotic hypothesis? So these are given in the 7 steps. So first we will discuss these steps. So first is ATC, that is the electron transport cycle. It is an oxidative phosphorylation reaction that takes place in the inner membrane of the mitochondria. So it will take place in the inner membrane of the mitochondria. It begins with NADP, it begins with NADH and FADH2, and it loses the electrons. So these electrons, they are transferred along the chain. So these electrons will be transferred along the chain. As each complex accepts electron and passes them on, energy is released. So energy is released when each of the electron passes them on. So this energy is used to pump the protons against the concentration gradient from the matrix of mitochondria to the inner membrane space. So there will be a energy which is used to pump the protons. Even actually there is a high concentration of protons. So there will be a high concentration of protons in the membrane space, and the protons, they try to move back into the matrix. However, the inner membrane is impermeable to the protons. Now the 6th point is a proton motive force is set up, and the ATP synthase undergoes a conformational change and uses the PMF to make the ATP from the ADP. Now here it is written ATP plus phosphate ions gives the ATP reaction. So ATP synthase allow the transport. ATP synthase is an enzyme, and we have already discussed the function of ATP synthase. So it will allow the protons to diffuse back down their gradient. So it will allow the protons to diffuse back to their gradient. So these are some of the references.